All right, everyone, we're going to begin. So it's my pleasure to welcome you to the webinar, Definition Through Consortium, a recap of the Sarcopenia Definitions and Outcomes Consortium. My name is Ryan Carney. I work in the Public Policy Department at the Alliance for Aging Research, and I serve as the manager for the Aging in Motion, or AIM Coalition. Before we begin, I'd like to provide some background on the AIM Coalition. So the AIM Coalition is made up of 30 nonprofit patient and research advocacy organizations working together to increase recognition of sarcopenia and age-related functional decline. AIM was founded in 2011, and our core goal is to promote clear clinical development and regulatory pathways for effective treatments that can improve the lives of people with sarcopenia. Our scientific advisory board are listed on the slide. We try to engage with industry and strategic activities to produce results that can be used in the diagnosis and treatment of sarcopenia. We also work directly with regulatory bodies in advancing our activities because we want to make sure that we meet their needs. I would also like to recognize AIM sponsors: Abbott Nutrition, Astellas, Astamed, Cytokinetics, Longeveron, Metabolic Technologies, Nestle Health Science, and Pfizer. Without your generous support, programs like this would not be possible. Thank you for your commitment. Today's webinar will be a recap of the Sarcopenia Definitions and Outcomes Consortium meeting, which took place on November 13th in Boston, Massachusetts. We are pleased to be joined by the meeting organizers for a presentation on the work that led up to the meeting, what transpired that day, and what the meeting means for the field of sarcopenia going forward. Questions will be reserved for the Q&A period at the end of the program, but as you come up with questions, feel free to use the WebEx chat function or email me at rcarney, that's R-C-A-R-N-E-Y, at agingresearch.org. Next slide. We're fortunate to have Dr. Jack Gorelnik as our moderator today. Not only is he the chair of AIM Science Advisory Board, but he also served as a Sarcopenia Definitions and Outcomes Consortium expert panel. Dr. Jack Gorelnik spent 25 years doing research at the National Institute on Aging, NIH, and was the chief of Intramural Laboratory of Epidemiology, Demography, and Biometry at the NIA. He also received multiple professional awards during his career. In 2005, he was awarded an honorary doctorate from the University of Tampere, Finland, and in 2009 received an NIH Director's Mentoring Award. He is board certified in public health and general preventative medicine, and he currently teaches at the University of Maryland School of Medicine. So Jack, did you want to take it over?
Yes, thanks, Ryan. Today we're really fortunate to have Dr. Shally Basine and Peggy Cawthorn, who have led the Sarcopenia Definitions and Outcomes Consortium for the past 6 years and served as PIs for the NIA and FNIH grants that have supported this research. Dr. Cawthorn is currently senior scientist at the California Pacific Medical Center Research Institute and associate adjunct professor at University of California, San Francisco. She earned her doctorate in epidemiology from the University of California, Berkeley. Dr. Cawthorn's current research interests include maintenance and mobility in old age, sarcopenia, frailty, the decline of physical function with age, and osteoporosis, particularly in older men. Dr. Cawthorn has several years of experience managing large multi-center observational studies. Our other speaker will be Dr. Shally Basine, who is professor of medicine at the Harvard Medical School and director of the research program for men's health and aging at the Brigham and Women's Hospital in Boston. He's also director of the Boston Claude D. Pepper Aging Research Center. Dr. Basine is really one of the foremost experts in men's health and aging. He's published more than 300 original research papers in top-tier journals, including the New England Journal of Medicine, JAMA, and the Proceedings of the National Academy of Sciences. He's conducted some of the most important randomized trials of function-promoting therapies for sarcopenia and functional limitations in older adults. Dr. Basine has been the recipient of numerous teaching and research awards throughout his career. He's been listed among Castle Connolly's top doctors in America every year since 2005. The Endocrine Society recently recognized him with the Outstanding Clinical Investigator Award. So we have two of the premier experts in this field who have led this effort at studying sarcopenia and working on the definition of sarcopenia over the past 6 years. And I will now turn the program over to them.
Hi, good morning. I want to thank the AIM Coalition for inviting Dr. Cawthon and me to share with you the outcome of the Sarcopenia Definition and Outcomes conference that took place in Boston on November 13. This effort was the culmination of 6 years of research and consensus building by this particular consortium. Next slide, please. The funding for the Position Development Conference was provided by a conference grant from the National Institute on Aging, the Foundation for NIH, and Aging in Motion Coalition. Additional support was provided by Astellas, Cytokinetics, and Abbott Nutrition. Next slide, please. So in today's presentation, Peggy and I will share with you some background on the historical evolution of this initiative, Sarcopenia Definitions, and how the consortium was established. We'll describe the analytical approach that was used to generate the position statements and the key findings of these analyses, the composition of the expert panel, the process that was used for developing these position statements, and the key takeaways from the meeting. Next, please. The term sarcopenia was initially coined by Evans, Campbell, and Rosenberg in the early 1990s, and these investigators conceptualized sarcopenia as age-related loss of muscle mass. Rick Baumgartner defined sarcopenia as the occurrence of lean mass 2 standard deviations below the mean for healthy young adults. And Newman reported that muscle strength depletion was more tightly associated with mortality than lean mass. From 2005 to 2018, various expert groups, national and international societies published multiple definitions of sarcopenia, and the European Working Group for the definition of sarcopenia has recently published an update on their previous definition. 2017 saw a major landmark development in the field when AIM Coalition successfully lobbied for and was able to promote an effort to establish the ICD-10 code for sarcopenia. So that now has enabled clinicians to use this ICD-10 code for billing purposes for sarcopenia condition. Next, please. So why is it important to have a consensus evidence-based definition of sarcopenia? An agreed-upon definition of sarcopenia is necessary to enable clinicians to diagnose patients in the clinical setting. And this definition would also be helpful in guiding the design of randomized clinical trials of function-promoting therapies. This definition can help operationalize the eligibility criteria in efficacy trials of function-promoting therapies and may serve as an outcome in some efficacy trials, similar to how bone mineral density has been used as an endpoint in some osteoporosis trials. Such a definition would also be useful to regulatory agencies in their review and approval of function-promoting therapies, and thus would be catalytic in supporting investments in emerging function-promoting drugs. Next, please. Sarcopenia meets Baker and Sprott's criteria for an ideal biomarker. Lean mass, muscle strength, and gait speed can be measured in a clinical setting with high level of precision and accuracy. Significant changes in these measures are demonstrable in clinical and experimental settings, and we know that from randomized trials of some of the function-promoting therapies such as testosterone. These lean mass, muscle strength are also responsive to anabolic interventions such as SARMs, myostatin antagonists, nutritional interventions, exercise interventions. In humans and species as far removed from humans as C. elegans, loss of muscle mass and strength is associated with poor outcomes, including mortality. Next, please. In 2009, Stephanie Studansky initiated a project to develop an evidence-based definition of sarcopenia, and this initiative came to be known as the FNIH-1 initiative. It was funded by the Foundation for National Institutes of Health. Several publications resulted from the FNIH-1 initiative, and then in 2013, the Sarcopenia Definition and Outcomes Consortium, or SDOC, was established to continue and extend the work that was started by Stephanie Studansky and colleagues in FNIH-1 initiative. There were several areas for which many investigators felt and funding agencies felt more work needed to be done. And this work, as I will explain to you in a minute, has been carried out by SDOC. In September of 2015, National Institute on Aging funded the SDOC Sarcopenia Cut Points grant. And in March of 2016, additional funding was provided by Foundation of National Institutes of Health for the Sarcopenia Definitions and Outcomes Consortium to extend the work that had previously been funded by NIA and to add some additional aims. Next, please. The SDOC Consortium has been led since its inception by Dr. Clausen and myself. We serve as the joint PIs for the FNIH as well as the NIA grant. The steering committee and key analysts include Tom Travison, Sheena Patel, Carol Pensina, Todd Manini, Roger Fielding, Jay Magaziner, Hao Su, and Rosalie Correa de Araujo. And a large number of investigators who are experts in this area have been involved in this initiative from the very beginning, and many others have contributed data, epidemiologic studies, as well as their input and expertise. Next, please. The SDOC Consortium differs from other efforts in this area. The SDOC project aims to develop evidence-based definition rather than opinion-based definition. Most of the published definitions that are out in the literature that have been published by various societies and organizations have been based upon opinion of expert bodies. In the SDOC effort, cut points will be derived from analysis of data from one of the largest assemblies of observational studies that have included large numbers of older adults with mobility complaints. SDOC 1 included community-dwelling adults, but had a very small representation of people who were functionally limited. The SDOC has evaluated these cut points against hard patient-important incident clinical outcomes that were deemed of public health importance. In addition to mobility limitation, these cut points have been evaluated against mortality, falls, fractures, and IADL disability. Additionally, the performance characteristics of these cut points, sensitivity, specificity, and predictive value, is being assessed in randomized clinical trials and clinical populations and in nationally representative population-based studies. Next, please. The SDOC project has assembled the largest collection of epidemiologic studies. It includes 10 observational studies of older adults that comprise close to 19,000 subjects, about 13,000 men and about 5,000 women, mean age 75 years. 17% of these older adults had mobility complaints. In addition, it includes 10 carefully characterized clinical populations from randomized trials and cohort studies in patients with hip fracture and HIV. There are 2 nationally representative population-based cohorts, the Health and Retirement Survey that includes about 4,000 men and women, 1,800 men, and about 2,200 women. The National Health and Aging Trend Survey, or NHATS, that includes about 5,600 men and women. Next, please. This slide summarizes the analytical approach, and you'll get a better sense of it when Dr. Cawthon does her presentation. We selected a comprehensive set of candidate sarcopenia markers. We harmonized the DEXA lean mass and some of the other outcomes. And as I'll explain to you in the next slide, the DEXA measures from each of the cohorts were calibrated to the NHANES standard. We evaluated the impact of body size using allometric scaling. We then used CART analysis as well as ROC curve analysis to identify cut points for candidate variables. These cut points then were evaluated in clinical populations and RCTs for their performance characteristics, sensitivity, specificity, and predictive value. These cut points and their association was then assessed in terms of their association with hard clinical outcomes such as mortality, falls, fractures, and IADL disability. Next, please. The DEXA data were harmonized across epidemiologic studies because the DEXA machines and the algorithms for deriving lean mass from these DEXA machines varied across epidemiologic studies. So the measures obtained on cohort-specific DEXA machines were calibrated to NHANES standard, which enabled harmonization of lean and fat mass estimates across studies. We also evaluated the influence of various coefficients of body size and adiposity using both ratio-based and regression residual approach. Next slide, please. I'm going to invite my colleague, Dr. Cawthon, to describe the results of these analyses. Peggy?
Great. Hi, Shelly, and thanks, everyone, for joining the call. I'll talk now about the method we used to identify variables of potential sarcopenia variables that discriminated those who had mobility limitation compared to those who didn't. So to do this, we used CART models, or classification and regression tree, which is a simple machine learning technique. A nice feature of CART is that it isn't limited by collinearity of variables. So you'll see of the number of variables we included, age, height, weight, body size, lean mass, a number of measures of strength, and these measures also standardized to body size. A number of these are very interrelated with each other, but CART isn't like linear models, which you may be more familiar with, where collinearity is a problem. So what CART will do, will pick the variables that most strongly discriminate the population based on the outcome. And so if we go to the next slide, you'll see what the results are. Next slide. So here is the results in men. The outcome here is walking 0.8 meters per second or slower. You'll see this was done in almost 14,000 men. And overall, the number of men in the entire sample who walked slowly was about 9%. The first cut point we identified was in grip strength divided by BMI of a value of 1.05. Those who had a value above this were much less likely to walk slowly. Only about 7% of this group walked slowly. Well, as those who had a grip strength divided by BMI of less than 1.05, these folks were much more likely to walk more slowly. About 30% of this group walked slowly. And then you'll see a secondary cut point was for age, and then a tertiary cut point in a subgroup of age was in arm lean mass. Next slide. We saw somewhat similar results in women in that the first cut point is for grip strength divided by a measure of body size, in this case, body fatness. And again, we were able to strongly discriminate groups of people who walked quickly from those who didn't. You'll see secondary cut points for women included age and grip strength divided by arm lean mass, and some further cut points for other measures of either fat and lean mass ratios or age. Next slide, please. We ran a number of different analyses in addition to CART, including some ROC analyses and CART models with different outcomes, including different cut points for gait speed, for example, 0.6 meters per second or 1.0 meters per second to find mobility problems. And we also used logistic models. And from these models, we found a number of consistent variables that were selected by the CART models to discriminate those who walk slowly from those who don't. The variable that was consistently chosen for men and women in many of these models was grip strength divided by BMI. And you'll see on this table a number of other measures of grip strength that were selected. The only measure that really came out as a first or second cut point in both men and women was leg lean mass. That's the only measure that didn't include grip strength. None of the other measures, including BMI or fat or height or weight, were primary or secondary cut points. So from these cut points that we developed in the CART and logistic models, we then applied these cut points to various outcomes that we collected in our cohort studies. So next slide. So the first outcome we examined was mortality. These are hazards ratios for mortality that have been adjusted for age. And the male results are shown in red rectangles, and the women's results are shown in blue X's. And you'll see that for many of the measures of low grip strength, either standardized to body size or fatness or lean mass or not standardized, that these having a low value is associated with an increased risk of mortality. The association between low leg lean mass for women was not significant. It was borderline significant for men. Next slide. And then the summary for the incident falls is fairly similar in that low measures of low grip strength, either standardized to BMI or not, were generally associated with an increased risk of falls. However, the association between low leg lean mass by this measure and incident falls was not significant, and it was suggested that it was maybe in the opposite direction as you would expect, such that low lean mass would be protective against falls. We would expect it to be a risk factor. So next slide, please. So this is just a snapshot of all of the results that we've developed, and we presented those summary results in a previous webinar for this group. But in general, all of the results taken together suggests that muscle weakness, when it's defined by low grip strength, is a predictor of adverse health-related outcomes, including mobility limitation and falls, ADL disability and mortality in older community-dwelling adults. I didn't show the results for disability or incident mobility limitation in the interest of time. In summary, also our results found that appendicular lean mass when measured by DEXA, either when you take appendicular lean mass in sum or when it's standardized to body size, for example, divided by height squared, it was not a consistent and good predictor of adverse health outcomes in our community dwelling population. And finally, we also found that both low grip strength and low usual gait speed independently predict these adverse health outcomes in older adults. So when we would include grip strength as a covariate and a model for gait speed and vice versa, both were significantly associated with those outcomes. Next slide, please. So we presented these results to our project team and a number of other guests at a meeting in October 2017. And how to make these results, synthesize the results into a definition or at least statements related to sarcopenia wasn't totally clear. So we came up with the idea of having a physician development conference shortly after that October meeting where we presented our results. And the idea about the physician development conference was to integrate and summarize expert opinion about our results and how they fit into the literature and what other working groups have done in the development of a sarcopenia definition. Next slide, please. So as you've heard today, we had this physician development conference just before Thanksgiving, and a number of members of this SDOC team presented the summary of results and a short background of the literature around a number of different areas. The presenters included Shally and myself, as well as Roger Fielding, Doug Keel, Jay Magaziner, Todd Manini, Ann Newman, and Tom Travison. The expert panel was selected based on international representation. We want to get people who had been involved in the development of previous sarcopenia definitions who weren't previously part of the analysis team for SDOC. And you'll see the expert panel listed here. Cyrus Cooper was the chair, Hidenori and Arari, Brian Clark, Jane Cawley, Jack Ralnick, Francesco Landi, Suzette Piera, Dan Rooks, Laura Schapp, and Jean Wu were all members. Next slide, please. So how did the process work for the Physician Development Conference? Well, over the summer, the SDOC team drafted position statements, and we presented this along with background information to the expert panels on a series of conference calls. The expert panels reviewed those position statements as they were drafted and suggested changes and additional background information or clarification about the analyses we presented. Then, based on input from the expert panel and revision from the SDOC team, we finalized the position statements. Then, at the November 13th meeting, the final background information and position statements were presented to the expert panel for a final vote. And this included discussion and input from both the expert panel and the audience members. And the meeting that we held was very interactive. We had lots of discussion. Several hours of the day were dedicated to discussion and raising different points. And I think the collaborative and really invested interest of people in the outcome was really useful for understanding the different viewpoints on some of the statements. And then the next step will be to publish the meeting proceedings and disseminate those results through efforts like this, through this webinar, for example. So the next step, or next slide, please. So how did the voting work? So the expert panel voted on each statement. And essentially, you can think of the expert panel as a jury that evaluates each statement. And they vote on each statement on a scale of 1 to 9, where 1 is strongly approved and 9 is strongly disapproved. And so uncertainty about a statement can be indicated in several ways. The first is that most or all members vote in the uncertain range of 4 to 6. And the second is that if there is disagreement on a panel, for example, some vote to agree with a score of 1 to 3, and some vote to disagree or disapprove with a score of 7 to 9. So next slide. And so the format, and I discussed this already to some extent, but we had 4 sessions. One was about grip strength, one was about lean mass, one was about gait speed, and one was a summary overall discussion. And basically what happened is the position statements and background information were presented, then the expert panel, and then the audience made comments, and then there was a voting session. Next slide. So here's a picture of what the discussion looks like. I like this picture because it really shows how intently the expert panel members were listening to each other and discussing, and that there was really a lot of collaborative and interesting comments made that will be reflected in our meetings proceedings paper. Next slide, please. So here's the first position statement and then how the panel voted. So our first position statement and the first 4 all are about muscle weakness, but the first position statement had strong agreement, and this statement was muscle weakness in older adults can be conveniently defined using grip strength. Next slide. Now here's the second position statement, and again, this had strong agreement from the panel. And this second statement was muscle weakness is defined by low grip strength, is a predictor of adverse health-related outcomes such as mobility limitation, falls, ADL disability and mortality in community-dwelling older adults. Next slide, please. There was a similarly strong agreement for our third statement, which was muscle weakness is defined by low grip strength should be included in the definition of sarcopenia. Next slide, please. And again, good agreement for our fourth statement about performance characteristics of a sex-specific cut point for low grip strength may vary by age, race, disease condition, and other factors. Next slide. Now, the next session for the meeting surrounded measures of appendicular lean mass. So our first statement here, appendicular lean mass by DEXA, either absolute or scaling for body size, is not a good predictor of adverse health-related outcomes in older adults. And you'll see here again that there was good agreement from the panel members for this statement. Next slide. Now there was, moderate to good agreement for this next statement that was that highly variable risk associations found between appendicular lean mass by DEXA and adverse health-related outcomes in community-dwelling older adults limits the utility of lean mass assessed by DEXA as a predictor or prognostic factor. Next slide. I just had my screen go blank. Hold on. I have to log back in. So then I'll try to talk through this while I'm... Okay, next slide. I see it here. Okay, number 7. Lean mass measured by DEXA should not be included in the definition of sarcopenia. Now, this is the only statement that elicited a fair amount of uncertainty, and you'll see the distribution of scores from the panel members is quite wide. And when we're done with going through the voting, I'll discuss this point in a little bit more detail. Next slide, please. So the next 3 topics were about slowness as defined by usual gait speed. And you'll see that the panel members had strong agreement that slowness is a good predictor of adverse related health outcomes in older adults. Next slide. And that the panel also agreed that strength is one of the many factors that influence usual gait speed. Next slide. There was also good agreement that usual gait speed is an indicator of walking ability should be included in the definition of sarcopenia. Next slide, please. The panelists also agreed that the performance characteristics of a cut point for low usual gait speed might vary by several factors, including age, race, sex, and disease conditions. Next slide, please. Now, the last two position statements that were voted on during the conference combine several of the domains together. So this last statement, the next to last statement here, statement 12, reads low grip strength and low usual gait speed independently predict adverse health-related outcomes in older community-dwelling adults. The one vote that was a little bit uncertain on this reflected the uncertainty of the lean mass statement and whether lean mass should be included in a sarcopenia definition based on discussions with Dr. Arari, who had that more intermediate vote. Next slide, please. And then this is the last statement that there was very good agreement on, again, that both weakness defined by low grip strength and slowness defined by low usual gait speed should be included in the definition of sarcopenia. Next slide, please. So to summarize the outcome of the position statements, you'll see that overall there was strong and uniform agreement on most of the statements that were considered. There is endorsement that both gait speed and grip strength are good prognostic indicators of outcomes in older adults, and that both should be included in a definition of sarcopenia. And reflecting the, the overall scientific community and discussion, there was uncertainty about the role of DEXA in a definition of sarcopenia. But I think highlighting the uncertainty surrounding that point identifies the areas for reaching a true, universal consensus on a definition of sarcopenia is really a step forward, in that people can now, refine their disagreements maybe around just the DEXA issue and not so much whether or not grip strength and gait speed are relevant measures to be included in a sarcopenia definition. So our next steps based on the meeting are to publish the results of the SDOC analyses and a meeting, a conference proceedings paper. And we've already been in discussions with the journals of gerontology of a submission like that. And then I'll hand this back over to Shally to summarize where this fits in the development and continuous evolution of definitions.
Thank you, Peggy. So the November 13 Sarcopenia Definitions and Outcome Position Statement Conference was a very major milestone because we were able to get some of the world's top experts who were independent of the consortium's work to review the position statements that evolved naturally from the analysis that the consortium had conducted. And you saw that on 11 of the 13 statements, there was very high level of agreement and high level of certainty. And there was a fair bit of discussion around the lean mass statement, but the expert panel agreed that lean mass was not a predictor. So this, I think, puts us in a very good position to build upon these findings and to then go to the next steps. In many ways, the development of consensus definitions and guidelines is a continually evolving process, and it's been the basis of progress in medicine. As an example, the first evidence that high cholesterol was a risk factor for heart disease emerged from the analysis of the Framingham Heart Study. And this analysis was completed in 1950 during the very first 5 years of the contract.
of Birmingham Heart Study that was funded by National Heart, Lung, and Blood Institute. The first ATP1 consensus treatment guidelines, which came to be known as the ATP1 guidelines, were published in 1985. And over the last 30 years, these guidelines have evolved in very significant manner based upon new data that has emerged from additional epidemiologic studies and randomized clinical trials of cholesterol-lowering drugs. And so in 2013, we had ATP4. Similarly, in the area of hypertension, the Joint National Committee for Blood Pressure Detection, Treatment, and Prevention, again funded by NHLBI, started its work in 1977 with publication of the JNC1 guidelines for blood pressure management. And last year, we saw the publication of the JNC8 guidelines. So, it's an evolving process. And the way we see it, SDOC 1 Position Statement Conference was really a foundational step in the evolution of sarcopenia definitions, and it'll evolve over time as more data becomes available from additional epidemiologic investigations, additional analysis, and from randomized clinical trials of function-promoting therapies, which will enable us to actually test these definitions in the context of randomized clinical trials, just as the randomized trials of cholesterol-lowering drugs and blood pressure-lowering drugs enable testing rigorously of ATP and JNC guidelines. So I think one important mandate that we received from the meeting is the need to sustain these efforts initiated by SDOC based on the ATP and JNC model to continue to refine the risk models and generate progressively updated guidelines for diagnosis, treatment, and prevention of sarcopenia in the general population and in older adults with specific conditions. And I think this work will be crucially important for the development of drugs for the treatment of functional limitations in older adults and in guiding clinical practice and health policy. So let me stop here and thank all of you for participating in this conference, and also thank the AIM Coalition and Jack Goralnik for inviting us to share these thoughts with you. Thank you.
Great, thank you. And we're now opening up the webinar for questions. You can use the WebEx chat function, or you can email me once again at rcarney, that's R-C-A-R-N-E-Y, at agingresearch.org if you have a question that you would like to put forth. Wait a moment. All right. Someone asked, how do you see the work of the consortium relating to the European Working Group's recently revised definition?
Yeah, thank you. That's a particularly relevant question since the European Working Group published an update of their definition. I think if you look closely at the European Working Group definitions that they published a few years back and the new ones, there's not a large difference between our definition and their definition. I think we were able to more concisely define this based on the analysis of data. So the position statements that have come out of the SDOC effort are based upon real analysis of real data. And so they are evidence-based as opposed to the European Working Group's effort, which were more opinion-based. Nevertheless, I think this type of conversation needs to take place, and then as more data becomes available, it'll enable us to harmonize the definitions so that clinicians can use a single definition This is just the natural evolution of knowledge and science.
Great. Another one just came in, says there were a number of disease-specific cohorts in the study. Will you be looking at them individually to see how your general cut points work in them?
Yes.
Yes.
Yeah, Peggy?
Go ahead, Sean. Yeah. We have looked at the how the different cut points work in them, and there are some variations, as you would expect, in the prevalence of slowness or low grip strength by the different conditions, and how the sensitivity and specificity of these measures for mobility disability also vary somewhat by disease condition. But also, how the definitions work in some of these cohorts is a little bit difficult to assess entirely. For example, the populations with hip fracture, immediately following the fracture, they're not mobile, and it's not clear how to classify people who are on a trajectory of recovery versus a decline. So I think there needs to be a little bit more work done in some of the disease-specific areas, especially those that don't fit with sort of the standard age-related decline measure, like the hip fracture cohorts, where we'd really want to do is measure an improvement.
And we know from our initial analysis of hip fracture and HIV populations that these cut points, as expected, their performance varies in different populations. And as Peggy mentioned, we need to do more analysis of much larger samples of patients with specific conditions.
Another question came in. How do the results of the consortium, or how do they inform the best method for screening or diagnosing the condition in clinical practice?
These position statements form the conceptual foundation of a quantitative definition that clinicians can use. But to build such a predictive risk model, we need more work to integrate these position statements and also take into account age, sex, and race. So our plan is to extend the work into its next stage, which will enable us to build a predictive risk model and will incorporate these position statements. But that risk model will be more quantitative to enable its use by clinicians as well as in operationalizing eligibility criteria in randomized trials. Peggy, your thoughts?
Yeah, I agree. I think in terms also this provides, I think a lot of people have maybe paused about whether or not DEXA lean mass is an important clinical component of a definition. There was definitely uncertainty in this area, and so I think that just highlights maybe something, a challenge to know how that could be operationalized, especially given challenges in, say, DEXA screening for osteoporosis, where there is clear consensus that DEXA-based BMD measures for osteoporosis are an important part of the diagnosis criteria, but there is screening and the number of people who get those scans, when there is frank agreement, is still very difficult. And so I think a careful consideration of DEXA in particular in a clinical setting is important.
One additional operational issue that came up during the discussion at the conference was the need to harmonize procedures. And several clinicians pointed out that the methods for measuring grip strength and gait speed are not, they vary sometimes a little and sometimes a lot across various studies. So one takeaway from the meeting was that the SDOC group will prepare a procedure manual, and that will harmonize the measurement of grip strength and gait speed to make it possible for clinicians to operationally implement the definition in their clinical setting, and also it'll make it easier for investigators and companies that have products in this space to operationalize it in the setting of randomized trials.
Fantastic. Another question came in. Said, do you see grip strength and gait speed to be independent to diagnose sarcopenia, or do you consider the combination of the two factors? Well, what would you consider to be the best primary endpoint for RCTs in the sarcopenic population for a clinically relevant benefit?
So this was an issue that was discussed quite extensively internally and also with the expert panel. And our analysis suggested that for some health-related outcomes, the slowness and weakness, when they were applied in combination, were better predictors of the outcome than either one alone. So hence the position statement that the definition should include both grip strength as well as gait speed. But one thought that was discussed and articulated by some of the panel members and people in the audience was that perhaps weakness represent an earlier stage. People may first be weak, and then they also develop slowness. And it's possible that weakness and slowness may represent different stages of the condition. But the data that we have didn't enable us to come to that conclusion based upon the analysis that were done. So I think given the results of the analysis, the statement that could be supported by the data was that both slowness and weakness should be included in the definition, and the two together are more predictive of some of the health-related outcomes than either one alone. Peggy, your thoughts?
I think that articulated the first part of the question very well. I think the second part of the question about what endpoint should be used for randomized trials, that's something that we didn't really address directly in our data or in the statement. There have been comments that regulatory agencies may not accept just strength and weakness alone as outcomes because they aren't necessarily patient-reported. And so how they're related to patient-reported outcomes isn't as completely clear. Also, our disability-reported, self-reported outcomes. There are associations, weakness and slowness associations with those disability-reported outcomes. So I think the endpoint for sarcopenia trials is a big point of interest for a number of people, and that's just not something that we received. The fact that we wanted to set the foundational issues first, and then hopefully in future conferences we'll address what endpoints are best for trials, for example.
Great. Well, that was our final question. So the recording of this webinar will be available on demand viewing at www.aginginmotion.org, and that'll be available within the next week or so. I'd like to thank our wonderful presenters, Dr. Cawthon, Dr. Bhasin, and our moderator, Dr. Gorelnik. I'd also like to thank our sponsors, Abbott Nutrition, Astellas, Astomed, Cytokinetics, Longeveron, Metabolic Technologies, Nestle Health Science, and Pfizer. And I want to thank everyone who joined us today for the presentation. So enjoy the rest of your afternoon.
Great. Thank you for the opportunity to present. We appreciate it.
Thank you.
Thank you.